Hello friends, welcome back to a new video, that is MCQs on biotechnology, and this is the fifth part of this series of videos on biotechnology. So the first question of this video, that is the term in situ is referred as, that is referred to option A inside body, option B outside body, option C in situation, or option D none of the above. And the right one is option A, that is inside body. So the term in situ refers to inside body. So let's move on to question number 82, that is the term in vivo refers to option A inside body, option B outside body, option C in viruses, or option D in vector. And the right one is option B, that is outside body. So the term in vitro refers to outside body. So next question देखेंगे, question number 83. So the incorporation of wild type gene into domesticated breeding lines or from transgenic into wild type genome is referred as option A gene conversion, option B introgression, option C gene filtration, option D gene therapy. And the right one is option B, that is introgression. So the incorporation of wild type gene into domesticated breeding lines or from transgenic into wild type genome is referred as introgression. Okay, so next is question number 84, that is genes that moves within genome is or are referred as option A mobile genes, option B parasitic genes, option C jumping genes, option D introns genes. So the right one is option C, that is jumping genes. So the genes that moves within genome is or are referred as jumping genes. So the next one is question number 85, that is protein oligomerization is mainly contributed by option A hydrophobic bonds, option B disulfide bonds, option C both A and B, option D none of them. And the right one is option C, that is both A and B. So the protein oligomerization is mainly contributed by both A and B, that is hydrophobic bonds and disulfide bonds. So the next question is question number 86, that is a study to compare functions of characterized gene of an organism to function of unknown gene of another organism is known as comparative genomics, metagenomics, epigenomics, or functional genomics. And the right one is option A, that is comparative genomics. So a study to compare function of characterized gene of an organism to function of unknown gene of another organism is known as comparative genomics, metagenomics, epigenomics, or functional genomics. And the right one is option A, that is comparative genomics. Okay, so let's move on to question number 87, that is human genome was sequenced by using which of the following approach. And the options are option A chromosome walking, option B cytogenic mapping, option C shotgun sequencing, option D radiation hybrid mapping. And the right one is option C, that is shotgun sequencing. So human genome was sequenced by using which of the following approach, that is shotgun sequencing method. So let's move on to question number 88, that is C value paradox is option A DNA content of haploid genome, option B RNA content of an organism, option C cellular protein content, option D lipoprotein content of plasma. And the right one is option A, that is DNA content of haploid genome, that is known as C value paradox. So the next one is question number 89. So which of the following statements are correct for genome in situ hybridization? And the options are option A whole genome is used as probe, option B it is used to study the evolution, phylogenetic relationship and divergence of different species, option C it helps in characterization of chromosome of breeding lines, option D all of the above are correct. And the right one is option D, that is all the above are correct. So So this all are the following statements are correct for genome in situ hybridization. Okay, so let's move on to question number 90. So the depiction of gene loci based on frequency of recombination in the offspring genome is referred as option A linkage genes, option B linkage map, option C mutant link, option D broken link, and the right one is option B, that is linkage map. So the depiction of gene loci based on frequency of recombination in the offspring genome is referred as linkage map. Next one is question number 91. The phenomenon of attaching a drug or a toxin to a monoclonal antibody or another homing molecule of immune system which is reversible is referred as option A homing, option B linking, option C joining, option D pairing, and the right one is option B, that is linking. So the phenomenon of attaching a drug or a toxin to a monoclonal antibody or another homing molecule of immune system which is reversible is referred as linking. So next one is 92. That is in 1970 monoclonal antibodies are discovered by option A Kaiser Milstein, option B George Kohler, option C both A and B, option D neither A nor B, and the right one is option C, that is both A and B is the right one. So the in 1970 monoclonal antibodies are discovered by Milstein and Kohler. Okay, so the next one is question number 93. Any crystalline structure possessing dimension measured in term of nanometer is called nanotechnology, nanocrystal, nanobiotech, supercrystal, and the right one is option B, that is nanocrystal. So let's move on to another question, that is the device that can differentiate between two DNA strands that differ in only one nucleotide to is referred as nanopore, option B nanobiotech, option C nanonucleotide, option D none of the above, and the right answer is option A, that is nanopore. So the device that can differentiate between two DNA strands that differ in only one nucleotide is known as nanopore. Okay, so that device is known as nanopore. Okay, so let's move on to question number 95. So the nanotech device called nanopore was firstly developed by David Demer, or option B Mark Atkinson, option C both A and B, or option D David Baltimore, and the right one is option C, that is both A and B is the right answer. That means David Dreamer and Mark Atkinson are right answer. So the nanotech device called nanopore was firstly developed by Dreamer and Atkinson. Okay, so let's move on to question number 96. That is reverse 2 hybrid system is used to determine option A function of protein, option B protein-protein interaction, option C role of amino acid residues in the protein-protein interaction, or option D protein structure determination, and the right one is option C, that is role of amino acid residues in the protein-protein interaction. So reverse 2 hybrid system is used to determine role of amino acid residues in the protein-protein interactions. Okay, so next one is question number 97. So primer extension is used for option A detection of transcript start site, option B expression profiling, option C RNA degradation, option D protein refolding, and the right one is option A, that is detection of transcript start site. So primer extension is used for detection of transcript start site. Okay, so the next one is question number 98. That is what is RNA editing, and the options are option A 5' capping, option B a mechanism to edit mRNA to get more than one protein product, option C polyadenylation, option D alternate splicing, and the right one is option B, that is a mechanism to edit mRNA to get more than one protein product, that is known as RNA editing. Okay, so let's move on to question number 99, that is the specific PCR technique where two consecutive run PCRs where second PCR amplifies further the product in first PCR is called dual PCR, option B hybrid PCR, option C nested PCR, option D RT-PCR, and the right answer is option C, that is nested PCR. So what is nested PCR? Where the specific PCR technique where two consecutive run PCRs where second PCR amplifies further the product in first PCR, that is known as nested PCR. Okay, so the last question of this video, that is the DNA structure has same sequence rate from left to right of one strand and right to left of another strand is called option A heteroduplex, option B palindrome sequence, option C heterodiploid gene, or option D consensus sequence, and the right one is option B, that is palindrome sequence. So the DNA structure has same sequence rate from left to right of one strand and right to left, that is left to right from one strand and right to left from another strand, and both are the same. So that type of sequence is known as palindromic sequence. Okay, so these are the all 20 questions of this video, that is part 5. Okay, part 5 of MCQs on biotechnology. Okay, so thank you for watching this video.